Hey, BioE 131. This is Russ with technical briefing number 9 about gene drives. A very interesting idea. So let's, in this module, I'm just going to give an example in the context of mosquitoes and malaria. And at the end, I will try to remember to broaden and open up this idea to the more large uses and implications. But let's start off with malaria. So malaria is a bad disease, and it kills 1 to 3 million people per year. And most of those people are kids who are less than 5 years old, and 80+% of those people are in sub-Saharan Africa. So by any standard, this is a big problem. And one that could easily invoke pretty extraordinary measures in order to save lots of lives. In order to understand what a gene drive is, I have to tell you that malaria is transmitted by a mosquito. And I think you probably all know this, and you know that we've tried nets, and we've tried insecticides, and we've tried other mosquito controls. Of course, we've also tried drugs to prevent and treat, and yet we have 1 to 3 million people dying per year, so we can declare those helpful but not solving. So the big idea for a gene drive is, wouldn't it be cool if we could either remove or drastically reduce the mosquito that transmits malaria. This is the Anopheles mosquito. So no mosquito, no malaria. That's the simple idea. As you can imagine, there are complexities to that. So remove or drastically reduce the mosquito population. Or, wouldn't it be cool if we could do that? Or, would it be cool if we could reduce the ability of the mosquito to transmit malaria? What do I mean by that? I'm just making this up, but you could make it not interested in biting humans. You could make it unable to host the malaria by changing its biology, etc. Use your imagination. You could do lots of things so that the mosquito was not an effective trim. You could stop it from flying so that mosquitoes just sit around, but they can't fly. Again, I'm making all this up. But whatever the idea, killing or reducing their ability, gene drives provide a way to do this that is really unprecedented. Now, as we talk about gene drives, there's a few concepts I want to review. One is the driven gene. In the context of a gene drive, so let me just write gene drive here. This is the gene that you are spreading rapidly through the population that is targeted, which in this case is the Anopheles mosquito. The idea is you find a gene that would make it infertile in the case that you want to remove or drastically reduce, or you identify a gene that would mess up its ability to transmit, as in case 2. So that's an important one. The next concept, which many of you will know from biology and genetics, is homozygous. Remember, we've talked about this, I think, before, that every living organism that has a mother and a father is typically homozygous, which means they have one copy from the dad and one copy of the genome from the mom. And therefore, for any gene, you're homozygous if the 2 copies are the same. Both copies are the same. And that contrasts with heterozygous, where mom and dad gave you different copies. They different copies, either slightly different or very different. And you'll see why this is relevant. So driven gene, homozygosity, and then the third concept that you need to know, which you do know, is CRISPR and guide RNAs. And I will refer you to a previous technical briefing if you have to bone up on that technology for editing genome using a guide RNA that targets and CRISPR that does the cut and then subsequent mutation, typically. Okay, so now that was a lot of setup, and I can tell you, let's discuss the example where we have a gene that, if we can knock it out, makes the fly infertile. So the case that we're going to study here is introduce rapid mosquito infertility. And it's going to be gene F, let's say. Okay. So how do we do it? It's actually scarily easy. First, we design a CRISPR guide RNA targeted to gene F to mutate it, yielding it losing its function, loss of function. So the guide RNA will introduce a break in the DNA, and there will be a repair, but that repair, in almost all cases, by the cell, will be an imprecise repair that introduces a mutation that mutates and renders that protein F with a loss of function. Now, you have it created. Now you introduce it into a population, a small population, so few in principle, of mosquitoes. You capture them, you introduce this so that it is in their germline. So in the germline, you're putting the CRISPR gene, because most mosquitoes don't have CRISPR. CRISPR is a bacterial protein, so you need to use CRISPR, and you need to put in the guide RNA, and you need to put in the necessary promoters so that both the CRISPR is expressed and translated, so you have active CRISPR protein, and so the guide RNA is transcribed so that it can then search around the genome, look for gene F, and make the change. So we'll do that into this population of mosquitoes. That's fine. Now you release this small population of engineered mosquitoes into the target geography. And I'm going to draw a picture in a second, but then what will happen is, I'm going to say step 4 is watch spread of mutated gene F in just a few generations to virtually all mosquitoes. So let's pretend that this is a mosquito. That's what we're going to call wild type, which means it's fully capable of transmitting malaria. And let's call the filled in circle an engineered mosquito. Engineered mosquito with the CRISPR setup that I just described. So to start out with, now here I get to draw a picture. At time 0, almost all the mosquitoes are wild type. At time 1, let's say we just introduce one of these engineered mosquitoes. Okay. What happens is this one will mate with a opposite sex mosquito. And what will happen? Well, what will happen is all of their offspring will be engineered. And I have to tell you why. Well, remember, this guy has CRISPR plus the guide RNA in both copies. They're homozygous in both copies of their DNA. So that was part of the setup. You did that in step 2. In step 2, you introduced this CRISPR and this guide RNA in a homozygous way so that they have 2 copies, which means no matter what copy they contribute to their offspring, their offspring will get at least 1 copy of the CRISPR and the guide RNA. The other copy will come from the other parent, but because the guide RNA and the CRISPR are there, that guide RNA will seek out gene F in the wild type and will edit it so that this offspring will eventually and very quickly become homozygous because they got 1 copy from this parent and the copy from the wild type parent was edited in real time by the CRISPR and the gRNA so that very quickly the offspring has 2 copies. And that will be true for all the offspring because they'll all get at least 1 copy of the guide RNA, and then they will do that. And then, I'm sorry, that's not very precise. And then they will all become homozygous themselves. So then what happens? This one of these, so that this, let's say, another one of these wild types will then mate with, and their offspring, same logic, will all be affected. That will happen here. They will all be affected, etc. Now, of course, if 2 wild types mate, their children, their offspring will, but eventually those offspring are likely to just by chance find mates. And depending on how big the initial population is, even with if only with 1 or 2 or a few, in a relatively small number of generations, all mosquitoes have the engineered dysfunctional copy of gene F. Now, as I said before, gene F could lead to fertility, and it could be either absolute or relative. That relative would be it's not absolutely infertile, but it's mostly infertile, or it could impair the infectivity and the ability to transmit, which I've already said this, and I recognize that. But I want you to see that what's happening here is that you're turning heterozygous individuals, genetically heterozygous, like these 3 offspring that I'm circling, they become homozygous. And that's what drives this very quickly through the population. Homozygosity in general will not be always, dominant traits alone. And a dominant trait is a trait that if you have 1 copy, it's enough for you to have that trait. You don't need 2 copies. This is a dominant trait, but it's kind of hyperdominant because it also changes the organism to become a homozygous organism. And so I hope you get that this means within a very short number of generations, everybody has the gene F. And then you just need to design your intervention, absolute or relative infertility, or impaired infectivity and transmission. And then this is a very exciting potential for controlling malaria. Now, zillions of ethical issues, and you'll read about those and we'll discuss them in the ethical portion of the class, but I wanted to make sure that everybody saw how relatively straightforward a gene drive is in principle to design and implement. Now, before we leave this discussion, I want to remind you that we were just talking about mosquitoes, but you could imagine gene drives in other organisms, including humans. So, for example, let's say we found some protein that makes, in fact, we have a protein mutation that markedly reduces cardiovascular risk, the chances of heart attack. They found this in the Amish population. There was a mutation in a gene where these families never had a heart attack. And it was remarkable, like virtually never. And it turned out to be a mutation in a particular gene that had a marked reduction in cardiovascular risk. So imagine that somebody says, cardiovascular disease is terrible. It's almost as bad as malaria. We should introduce a gene drive into humans so that everybody in a few generations will have low cardiovascular risk that will save hundreds of thousands, if not millions of lives, not to mention the money it will save. But as I hope you see, there are huge ethical issues there. However, this is a technical briefing, so I'm going to stop.